Okay, so in addition to mutation having an effect on natural selection, there's other things that have an effect on evolution. So one is gene flow. And if we remember a little bit about the gene pool are all the genes that are in a population right now. Gene flow means that you get new genes coming in or new genes going out. Genes are flowing in and out of the population. If you have new genes coming in, it could potentially change the population. You could get new gene combinations from the members that are coming in or out with gene flow. Okay, so for example, it's a pretty simple concept. If we had a field of mice here and a field of mice on the other side of the street, and they can't get to each other because there's a big street and nobody wants to cross that as a mouse, what we might see is that on one side of the street, you might have small and medium-sized mice. On the other side of the street, you might have medium and large-sized mice. If Palos Hills decides we're going to cut off that street so that we can have more nature, and now the mice can go back and forth, what we've done is we've created gene flow. The small medium are going to be able to flow into the medium large and medium large into the small medium. And so now what we have is we have more variability or diversity in the population, because on the medium large we've never had small mice, and on the small medium we've never had large mice. So now we have, the two populations are becoming similar. They both had medium. Now they're both populations where they were different, one population having that small genotype, or sorry, I should say phenotype, and the other population having the large phenotype. Now they both have small, medium, and large. Another thing that can happen is called genetic drift, which means that the genes are drifting away. So our gene pool is drifting. This doesn't have a huge impact on a large population. If you have a large population, likely you have a lot of variability. But if you have a small population, let's say that in a population you have emigration for whatever reason, a small group from a population decides to emigrate or move away, is it likely that that small population has taken every gene variability in that population? Probably not, right? And so you don't have as much variability usually in a small population. So genetic drift is going to have a greater influence on a small population rather than a large population. And I'm going to explain to you how. Okay, so let's say that in one small population. So what we have here is we have 20 individuals. And of those 20 individuals, we have 3 different genotypes. So we've got the capital B, codes for the black phenotype. You've got capital B, little b, codes for also the black phenotype. And you've got the little b genotype, which codes for the beige phenotype. So in this population, let's say something happens. There's a disaster that happens to this population, and the only 2 that survive is this one, big B, little b genotype with the black coat, and the little b with the beige coat. So these 2 survive, they mate, they make the next generation. So in the next generation, and kind of a little bit back about genetics, if these 2 are mating, the way that we would do that Punnett square, and in this case we're talking about complete dominance type of inheritance, is that you're going to bring this big B and the columns down. You're going to bring this little b and the columns down. You'll bring this b across and this b across. So here are all the possibilities of the genotypes that we can get in that population. So you can see in there that if they have 20 children or 20 offspring, that you can see that half of the offspring are going to have the big B, little b genotype and be dark in color for their coat, and the other half will be little b, little b. Let's say that something happens again to this population now. So in this population, the only two that survive are little b and little b. And if they mate, the only genes that they have are little b and little b. So in 3 generations, what you see is that you have 75% in the original generation has dark coat, 25% has light coat. If you have genetic drift and you only have a few surviving to the next generation, in 2 generations, you can get a completely different representation of the original gene pool. Another way to look at let's talk about causes of genetic drift first. There's 2 causes of genetic drift. One is the population bottleneck, and the other one is the founder effect. So first, let's talk about the bottleneck. With the bottleneck, we're typically talking about a natural disaster. We're talking about hunting, talking about loss of habitat by humans. So some may be human influenced and some may be weather or natural disaster related. What we see typically is that a very small portion of the population survives, and very likely that you're going to get a huge reduction in genetic variation. So you guys can't see it because your illustration below is in black and white, but what we see here, if it were in color, is that you see that you have a variety of 4 colors in the population representing 4 different phenotypes. And let's say something happens where only a small portion of what's in here survives. So randomly, what they did in this experiment was they just took this bottle and they poured it over to have a small number of individuals survive. And when they did that, what happened was only 2 of the 4 phenotypes survived. And so here again, you can see from the original population, you have a diverse gene pool. You have something that happens, some kind of catastrophe, hunting, human influence, maybe reduction in habitat, and only a small portion of the original phenotypes survives. And so we have a reduction in half of the different kinds of phenotypes survive to the next generation. So that's what we call population bottleneck. One example is the northern elephant seal. Back in the 1800s, they were hunted to nearly extinction, and only 20 individuals remained in the population. Of those 20 individuals, they had very similar phenotypes or gene pools. And so when they tried to protect them and increase the population size, it increased to 30,000 individuals. But here's the downfall, is that they're all very similar genetically. So now if something happens to this population, the likelihood that if the environment does not favor their genes or their phenotypes, their characteristics, then it could wipe out that entire 30,000 individuals. So second thing that can influence genetic drift is the founder effect. So here in the term founder is that you're having individuals who are in a small portion of the population decide to emigrate or move somewhere else, and they're all very similar. So they're bringing only a small portion of the genetic variability into this new population. They're founding a new population, but oftentimes that is not very good. So in this just little difference here is kind of more of the group moving rather than in what we're talking about the population bottleneck, that something happens to limit the population size. So we're lowering the frequency of the different genes in the population. So one example, it's a disease called Ellis van Creveld. This disease, some of the characteristics are that individuals will have an extra finger, which is, I don't know, I would think it was kind of a good thing, right? So in that, their gene pool is actually taking with them a beneficial mutation. However, one of the other things is that usually the torso is shorter, and so the organs don't have as much room, and there's often a lot of issues with, if we think about like the organs, the main organs of the body are in here, and so they end up having a lot of health issues. So here you have two big kind of phenotypes, right? Having that extra finger would be great because you would have a better grasping ability. But if you have this shorter torso, then you're going to have a lot of physiological issues over time. All right, so let's answer this question. Why does genetic drift affect a small population more than a large population? So give yourself a chance to read these. And what do you think the correct answer is? Good, B, yeah. So in B, chance event eliminates alleles from the small population, leaving it with a reduction in allelic variation. Good, very good. All right, so here we're going to focus on equilibrium population. This is going to go along with the lab today when we talk about Hardy-Weinberg. So do you think there's ever a case where evolution does not occur in a population? Meaning that there's never mutations that happen in a population. No, right? So we talked a lot about last time how when a cell is trying to copy all of the genes into a new cell for either sexual or asexual reproduction, that likely it's going to make some mistakes. So you know, we're going to look at some of the characteristics of what happens in a population, and we'll see that regardless of anything else that we can control, and we're going to look at factors that we can control in a population, we cannot control the copying of the genes from one cell to the next, meaning that you cannot control mutations. Okay, so we're going to take a look at all of these would have to be met in order for there to be no change in a population. So one, we just talked about, there can't be any mutations. We know we cannot control that. However, if we look at some of the other characteristics we could control, let's say in a lab setting, if we controlled all of the organisms that we were working with, we could control who mates with whom, who has contact with whom, right? You could control the flow of genes in that population. You could control the size of the population. You can control who mates with whom and when and how. You could control the environment so nobody in the population is favored based on their genetics. So we could potentially control 2, 3, 4, and 5, but we cannot control 1. So what we know for any population, evolution is always happening. We're going to look at how could we use mathematics to support this idea that evolution is happening and looking at changes in genes, just like we've looked at here, over time. Remember that if you have a large population, that the gene pool is probably very diverse, whereas a small population, if it's very small, the gene pool only may represent a few of the variations of the larger population. So again, we can control 2, 3, 4, 5, but we cannot control 1, which is mutation. Because we can't control mutation, evolution is always occurring or happening in a population. Oh, sorry. And last point again, just to reiterate, remembering that mutations happen by chance, they're spontaneous, just an accident in the copying of the DNA from one cell to the next. Remember again, mutations do not happen in anticipation or knowing what's going on in the environment. The cell can't say, oh, based on what's happening in the environment, it would be so much better for my offspring to have these traits. It doesn't work like that. Mutations are just by accident. All right, so we're going to take a look at the Hardy-Weinberg principle. This is here where we use mathematics to support changes in gene or allele frequencies over time. So if you remember back to kind of the first lecture, we looked at different things that can support the theory of evolution, those different kinds of data. Here's another way that we can use data to support evolution is happening. So in this, we're going to look at changes in allele frequencies or changes in the percentages of different genes over time to show what's happening right now and maybe what's happening in 100 generations or a long period of time in relationship to the generation time of the offspring. So if we're looking at bacteria, we could do this in maybe like a week to show a long period of evolution in those bacteria. But if we're looking at humans, we might have to take this data for like 1,000 generations before we could show this. What we're going to use is the kind of inheritance called complete dominance. This was an example that I used for complete dominance. You have a dominant allele or gene that masks the presence of the recessive allele. So that's why this still looked black in color and not a blend of the black and the beige, because this gene for black is masking the presence of the gene for tan, but that gene still does exist and is passed on. The only individuals in complete dominance who show the recessive trait or characteristic are those that have the two recessive traits, two recessive genes. So we're going to use Hardy-Weinberg. There were two scientists, Hardy and Weinberg. They came up with equations to show how we can do this. How can we show changes in allele frequency over time? The two equations that you're going to use are p squared plus 2pq plus q squared equal 1, and p plus q equal 1. I'm going to get into what the p's and the q's all mean now. So I'm going to walk you through this. P squared is the two dominant alleles. The genotype capital T. 2pq represents the heterozygous or one of each kind of allele, the capital T, little t genotype. And the q squared is the two recessive alleles, little t, or the little t genotype. P represents the percentage of the dominant alleles in the population, and Q represents the percentage of the recessive alleles in the population. So remember, a genotype is made up of two genes. An allele is one gene. All right, so let's get into P and Q again. So P squared is the frequency of what we call, again, the homozygous HOMO means same. P squared represents the homozygous dominant genotype. 2 genes, capital T, the 2 dominant genes or alleles. When we interpret the genotype into a phenotype, genotype are the 2 genes, the phenotype is how something looks. That when we interpret this, capital T, into a phenotype, those individuals look dominant. They show the dominant trait. It's the only thing they can show, right? They have 2 dominant alleles. PQ, it's heterozygous genotype. Hetero means other. That means you have one of each of the alleles. So that genotype 2PQ, capital T goes with the P, little t goes with the Q. We're going to have 2 of those. So in our equation, when we said P squared plus 2PQ, we have 2 heterozygous individuals. Because we're talking about complete dominance, as long as you have a capital T, it will mask that recessive allele. So these individuals will also look dominant. No blending here. This is complete dominance inheritance. And the last genotype is going to be the Q squared. Q squared is homozygous recessive. Homozygous means 2 of the same. Recessive tells us it's 2 of the same recessive alleles. So our genotype is little t. Because there's no gene to mask the recessive trait, 2 recessive alleles will show the recessive trait. So these individuals are the only individuals who will show the recessive trait or the recessive phenotype. Now, one of the things in genetics is that there's not that many traits that are controlled by one set of genes or that are controlled by one set of genes that exhibit the complete dominance type of inheritance. So you have to be very specific in what you're looking at in a population because there's just not that much complete dominance traits. All right, so we're going to take a look at also the alleles. Remember that P, anytime you see P, P represents the dominant allele of big T in this population. Q represents the recessive allele little t in the population. Two genes make a genotype, which code for a phenotype. Otherwise, one gene just tells you the percentage of that gene in the population. So we're going to take a look at frequencies of alleles now and on a long time from now or many generations from now and see if there's changes. And then that mathematic equations, those can support that evolution has happened in this population. So again, a lot harder with humans, right? We'd have to keep passing on the data every generation. Generations after 3 or 4 die off. So we'd have to keep it going for a long time. But if we looked at bacteria, it'd be easy. In a week, we could write down all of these pieces of data and calculate it very quickly. Okay, so I'm going to go through an example. This is going to be very similar to what you're going to calculate in lab today. So the data that you're going to start with, one piece of data. tells you 16% of the population exhibits the recessive phenotype. So let's think about what percentage of the population exhibits the dominant phenotype. Good. Okay, so of 84%, the reason why we don't start with this is because this is a little bit ambiguous. This 84% accounts for two possible genotypes, right? So 84% is made up of that or that. And just knowing this piece of data just tells us, okay, so if 16% of the population is the recessive phenotype, do you know the recessive genotype? What is the recessive genotype? Yeah. So we know the recessive genotype. This 84% is ambiguous in that we're not really sure what percent of that 84% is this or that. So we always start in Hardy-Weinberg with the recessive phenotype, because once you know the recessive phenotype, you know the recessive genotype. Here, there's too much ambiguity to tell what percent of that 84% is this or that. Okay, so based on knowing, if we know this, we know this, let's work on our equations. All right, so hopefully now you'll get this question right, that I went too far. I should have asked you first. So 16% as a decimal. Okay, so I hear Q and Q squared, which is good. You narrowed it down to the two possibilities. Remember that when we have the two equations for Hardy-Weinberg, not genotypes. Sorry. This equation is about genotypes. That when we use p squared plus 2pq plus q squared, we're representing the possible genotypes in the population. That p squared is big T, 2pq are two heterozygous or big T, little t, and q squared, which is our 16%, is going to be little t, little t. This other equation, when we have p plus q equals 1, we're talking about the percentage of the alleles. Percent of the dominant alleles is p, percent of the recessive alleles is q. So correct answer then, that 16% gives us q squared. So now we're going to keep track of this on the board as we go through the example. So if we're saying that 0.16 is q squared, we know this in our calculation. So now, since we know that, if Q is 0.16, we can take the square root of that to get Q. And so, Q is the square root of Q squared. So now we're going to take the square root of 0.16, and that gives us 0.4. So now we know that. And since we know this, we know Q, can we easily figure out P now? Yeah, right? Okay, so we can rearrange. I gave you the answer already. I'm going ahead of myself today. I'm all over the place, sorry. So if we rearrange this equation, and we change this equation into 1 minus Q equals P, our equation is 1 minus 0.16. What's P then? What is it?
0.6, because Q is 0.4.
Thank you. Okay, who wants to teach today? 1 minus 0.4 gives you, I heard 0.6. Okay, thank you, thank you. You guys are keeping me on my ball today. So now we know 0.6 and 0.4 here. So now we have all the tools to finish our calculations up top, because we know P and we know Q. So let's figure out P squared. So P squared is just 0.6 times 0.6, right? Or 0.6 squared. So when we take 0.6 times 0.6, we get 0.36. And we also, again, we know 0.6 and 0.4. So you can figure out 2PQ. So look at that. I didn't give you the answer for once. So 2PQ, 2 times 0.6 times 0.4 gives you which of these?
48.
Yeah, good. So 2 times 0.6 times 0.4 is 0.48. And now we've solved both equations from knowing one little piece of data. One thing I want to emphasize, and this is where a lot of students get stuck, is that the 2PQ represents 2 of the heterozygous genotypes. And I'll show you why that's important and how that's important. We can check our work. You guys should check your work to make sure after you do your calculations in lab that both of these equations equal what? Good, 1, right? So we're going to make sure. You might get, because you're working with real data, and I made up just perfect data to work out to exactly 1, but you might get like 0.99 or or 0.98 because you're only maybe working to 2 decimal places. So no, if you get like really close, within a couple of decimals, meaning like 0.01 or 0.02 on either side, maybe you get 1.01, then you're good. It doesn't have to be exactly perfect unless you're working with like 8 decimal places, but that's okay. So we want to be at 1 or really, really, really close. All right, so again, what's it got to do with evolution? We can take this data today, look at the frequencies today, look at the frequencies again many generations from now, see if there's a change in the allele frequencies over time. If there's a change in the allele frequencies over time, we know that that population has evolved. So back to kind of big concepts, remember that evolution is a change in gene or allele frequencies over time. At the core of evolution are genetics. You get your genetics from reproduction, and we know that the environment influences what genes or traits or alleles or characteristics are favored in the population. So back to Darwin and Wallace's 4 postulates. Those are the big concepts. Okay, so one thing I want to do before we move on I want to use this data and show you in a Punnett square where we would put all of the data. All right, so in a Punnett square, we'd put P squared there. We'd put, remember there's 2 PQs, so you get 1 PQ here and the other one goes there. And your Q squared goes there. In terms of allele frequencies or percentages of alleles, those would go out here. You'd put a P here, and a Q here, and a P here, and a Q here. So let's start with this equation. This equation is inside. This equation goes outside. So this equation, our data is going to go here. So we said 0.36 and 0.16. This 0.48, you don't put a 48 in each of these. You have to divide that by 2. 0.48 is 2 PQs. So we're going to divide that 0.48 and put half in each of the PQ squares. So these 2 squares are your 2 PQ, remember that. Your P plus Q equals 1, we're going to use this equation on the outside. So our P is 0.6, and our Q is 0.4. So let's check our math and make sure it works. So we checked it to equal 1, right? And so what I want to make sure is that my P and Q equal 1. That looks good, right? Again, my P and Q up top equal 1. Can check our math again. We do, remember, these go down, these across. So the way that we got 0.36 was 0.6 went down, 0.6 down. For 0.24, 0.6 down times 0.4 across equals 0.24. 0.4 goes down times 0.6 across equals 0.24 again, and 0.4 times 0.4 equals 16. When I add all of these up, so all 4 numbers in the big square, add those up, and they do equal 1 also. I want to talk about 2 more pieces of data we're going to get from this. Is that one of the first pieces of data we were given was that 0.16 was the percentage of individuals in the population that look recessive, right? So this when I ask you what percentage of the population look dominant, do you remember that? We look at all 3 of these. These 3 squares added together look dominant. 0.84 represents the addition of big T plus big T. So there's lots of data in this simple Punnett square, right? So this is kind of good data to have to refer to in lab today. As you're going through, you'll get equations. So we're going to work with our data in terms of our recessive traits in our population. So we're going to use raw data, meaning we're going to collect it today, and then we're going to figure out how many in our population have the recessive trait. And then you're going to go back and you're going to plug in, okay, I know this. So then from that, I'm going to get that. Once I have that, I'm going to take 1 minus that to get that. And then when I have that, I'm going to times it by 2 to get that. And then I'm going to times my P and Q times 2 to get that. And then you're going to fill this in. And then you're going to fill another table in that will ask you what percent show the dominant trait, what percent show the recessive trait, what percent of alleles in the population are dominant, what percentage of alleles in the population are recessive, what percent of alleles in the population show the homozygous dominant trait, what show the heterozygous trait, what show the recessive trait. Okay, so we're going to use all of that data to work with to remind us what all of this means. All right, we're going to do a little bit of